Today, I'd like to tell you what I'm not going to be talking about. First, I'm not going to be talking about charismatic megafauna like the Sierra bighorn sheep shown here. But what I am going to be talking about is charismatic and iconic megaflora, the giant sequoia tree. Most of you that approach a giant sequoia tree stand up and look upwards towards the sky. But what I do is I look downward in the soil beneath, the hidden half, and ask questions about how the plants affect the soils underneath them. Now, specifically, my research interests are how do plants shape the soils beneath them and that sustain them. And we know a lot about organisms that we observe above ground, their biology, their ecology, but we know comparatively little about the organisms that inhabit the soil and how they interact and affect the plants above them. Plants can affect soils in a variety of different ways. One way is by selectively taking up nutrients from the soil, and this affects the chemistry of the remaining soil. But they also can affect the soil through the deposition and senescence of plant litter, both leaves and roots, that then can impact the properties of the soil. Now, the interactions between plants and soils have fascinated foresters and ecologists alike for centuries. As early as the 18th century, Russian foresters already recognized that the success of the trees depended on the soils beneath them. As early as the 20th century, foresters started talking about beech soils or oak soils in reference to the unique soil properties that were developing under these different tree species. We were fascinated by the sheer lack of soil-plant relationship studies with the most iconic plant and the largest living organism, the giant sequoia. We hypothesized, given its sheer size, we're talking height as high as one football field high, and longevity, living millennia, up to 3,500 years old, that it would commensurately have a big effect, a ginormous effect, on the soil underneath it. Now, giant sequoia is in the family, the plant family, the cypress family, and there's about 140 or so species within that family. It's also in the subfamily redwoods, and there are only 3 species, of which it is one, that are in this subfamily. The coastal redwood, which it is most closely related to, which boasts the tallest tree on the planet, and also the dawn redwood, which is endemic to China, and it's a deciduous redwood tree, shown here in its full fall color. Now, the distribution of redwoods historically spanned the entire northern hemisphere. And the oldest fossils date back around 200 million years ago during the Jurassic period. But today, we are left with relic groves spanning a footprint about the size of the city of Cleveland on the western slopes of the Sierra Nevada mountains. And those are shown right here. So what shapes the distribution of these relic groves, at low elevation, it's lack of soil moisture. And at high elevation, it's legacies of past glaciation that occurred and ended, I should say, around 11,500 years ago. So in order to evaluate the imprint of giant sequoia on the soil, we need to compare it with something. We need to have a control. And so we selected another, I would argue, iconic species, the sugar pine, because it co-occurs with the giant sequoia in close proximity, and it also is very large and long-lived, up to several hundred years. In fact, naturalist John Muir called it the king of conifers. And it is only exceeded in size with other associated species with the giant sequoia itself. So we conducted our study in 2 of the 3 groves that occur in Yosemite National Park. And both of these groves experience Mediterranean-type climate with warm, dry summers and cool, wet winters in the rain-snow transition zone of the Sierra. The Merced Grove is slightly lower elevation, so it's slightly warmer and a little bit drier. However, the Mariposa Grove to the south has a little bit more water, a little bit more rainfall, and is slightly cooler. Now, they occur on two contrasting geological substrates. The Merced Grove, the soils are derived from metamorphosed sedimentary rock, and the Mariposa Grove, those soils are derived from a metamorphosed volcanic rock. So we used a paired experimental design where we selected 16 mature giant sequoia trees in each grove and then paired them with an adjacent sugar pine tree, shown on the right here. And then we ensured as much as possible to remove any potential confounding factors, such as their slope position or their aspects, or the presence or absence of understory vegetation, to make sure that we were studying the effects of the tree species itself and not some other confounding factor. Then, in August 2013, we sampled the mineral soil, the top 5 centimeters of it, and brought this mineral soil back to the lab and conducted a variety of different physical and chemical analyses that are known to affect plant growth. We also extracted the DNA from these soils and, using modern genetic methods, ascertained the differences potentially in the soil microbial communities that reside in the soil underneath the tree crowns. Well, this table here shows you some of the study characteristics of our trees. First, you'll know that, as its name attests, giant sequoia are indeed giant. They're about twice the diameter as the sugar pine trees in each grove, but more importantly, they're way more massive. Just the single stem of a giant sequoia tree has about the equivalent mass of 130 VW bug beetles. Okay, so it's pretty big. But, you know, the sugar pine's stately in its own right. It has an equivalent mass of about 30 VW beetles. So not too bad. So it made a pretty good comparison between the two. So the next two slides that I'm going to show you have similar format. The soil data from under the giant sequoia are shown in green. The soil data from under sugar pine are shown in red. The data from the grove, the Mariposa Grove, is on the left-hand side, and the data from the Merced Grove are on the right-hand side. So as you can see, the bulk density of the soil underneath giant sequoia is quite a bit less than it is under sugar pine. And this is due to the higher soil organic matter content under giant sequoia trees. Now, this is significant because it affects other properties, other physical properties of the soil as well, including the ability of the soil to breathe, its soil aeration. Also, its ability to store water, very important in a Mediterranean-type climate. So from all these different attributes, we see that the physical properties underneath giant sequoia are improved relative to sugar pine. Now, not only are the physical properties enhanced, but also the soil fertility. Soil fertility is improved dramatically under giant sequoia compared to adjacent sugar pine trees. And to demonstrate this with one piece of data, available nitrogen, which tends to be the most limiting nutrient to plants, is 40 to 80% higher under giant sequoia trees than under sugar pine trees. And indeed, when we analyze all of the nutrients that can contribute to soil fertility that are essential for plant growth, calcium, magnesium, potassium, in all cases, they were enhanced under giant sequoia trees relative to sugar pine. So we can conclude from this, just like the physical properties, that the chemical properties are improved under giant sequoia trees relative to sugar pine. Well, what about the biology? Well, using the extracted DNA, we were able to identify over 6,000 different species of bacteria and archaea. Now, this is an envy of anyone who studies biodiversity above ground. It's much greater below ground, my friends. And interestingly, there was no difference in the number of species, bacterial or fungi, between sugar pine trees and soil under giant sequoia trees. However, the bacterial membership in these communities differed significantly. In other words, there were novel bacteria that we found under giant sequoia that we didn't find under sugar pine, and vice versa. And to try to let you visualize that, I produced this graph here that us ecologists love when we're looking at multivariate data. And it shows that each of these symbols denote the membership of the microbial communities in the soils of these different groves and under the different species. And you can see that they all are different from each other, both by grove and by tree species that overlies them. But what's interesting is that there was this similar shift in the microbial communities from what we observed them to be under sugar pine to what they were under giant sequoia. And this similar shift in each grove suggests that the tree species are really controlling the composition of the bacterial communities that underlie them. Now, I could show you the same data. It looks almost the same for the soil fungi, but I'll spare you with that. But I will say that we identified a mere 200 species of different bacteria. So taken together, what the biological fingerprint says is that the tree species have a huge effect on not only the physical properties, the chemical properties, but also the biological community that they support and that the microbial community supports the tree. So understanding the interactions between organisms and their physical environment is what defines the field of ecology. However, we know so much more about the interactions between organisms that we can observe readily above ground than we know about the complex interactions that occur between soil microbes and plants, per se. However, with the advent of molecular tools, we're now getting an idea of what these unseen organisms, the bacteria and the fungi, how they can affect in positive ways our own human health, the so-called human microbiome. And it is very likely that such interactions also occur with the stately and iconic giant sequoia tree and the microorganisms that it supports in the soil. So next time you go into a giant sequoia grove and you look up and you're amazed at its sheer size and the influence it has on the above ground environment, turn your head downward and ponder equally the magnificent effects that these trees have on the below ground community. Thank you.